Thanks a lot to all the speakers. I have a few questions, and then we'll open it up for audience questions. I think we have maybe 10 minutes or so for discussion of these interesting talks.
Mike, can I make just before people leave, because some people are very hungry, at the end of the session, please all stay here. We're going to take a panoramic picture of everyone. Folks going to come inside, remove the table, and we're all going to come here.
So thank you for the announcement, commercial announcement. So open this up for everybody. What's the best endpoint for biomarker studies for immune therapy in the front line? And I just want to point out that for example, the axiabelumab trial that we just heard about had a PFS that was identical to the axi-pembro trial, but the overall survival results as reported were dramatically different. Now, we could talk at length about why that's the case, but clearly we would hope that the biomarkers we select might be associated with some sort of durable benefit, and PFS may be influenced by different factors. So what do you guys think?
I would say, some sort of definition of remission. So alive, small amount of tumor, maybe no tumor visible, on or off therapy at some distant time point.
Sabina?
Well, I mean, we have discussed this at length. I think in general, the immune-related PFS might not even, be the best biomarker if the median immune-related PFS. You're suggesting that maybe we should be looking at durable responses, so maybe more of a landmark PFS in the context of immune-related criteria makes more sense. Maybe an immune-related PFS at 12 months, 24 months.
Yeah. Other comments from any of the speakers?
I think in the absence, I think you have to tailor to each treatment. Overall, I would love to see overall survival, but this is not in the era of renal cell cancer therapeutics. This is not possible. I would love to see CR. We know they're durable, and even with VEGF-TKI, despite they're rare, they tend to be very rare, durable. But that's not possible. Based on that, I think a landmark analysis, because after 2 to 3 years, recurrences are low, or treatment-free survival, which you've been involved with Meredith and David, may make the most sense here. The issue is when you start combining and having 3 drugs or 4 drugs, that's the march of oncology. It works, you add it. How are we going to tailor to the biomarker that we established before?
Well, I think that the overall survival endpoint is what we want, but if you have the opportunity to cross over to other drugs that have survival benefit, then you're really not measuring the regimen you have. And so durable PFS or something, durable landmark ability to get a response and stop treatment is really what we're looking for.
And I think quality of life in combination with overall survival. And in the Ipi-Nivo versus Sutent trial, there was a difference. And I do think, and talk to my patients about that, if taking a pill every day and those side effects versus immunotherapy and coming for infusion. So
just one more question, then I'm going to open it up for. Did you want to comment on that?
Yeah, Mike, on that point, I thought you will lead the group and the audience to what you've been preaching, which is really treatment-free survival. At the end of the day, what patients want, and we have patients and patient advocates here, is really the ability to achieve a CR that's durable and stop therapy. And this was really what high-dose IL-2, obviously small number of patients are cured, but this is really the paradigm that we need to really use as the measure, as the yardstick. Can patients get CR that's durable, and then can they stop therapy? If we're going to treat them forever, I don't think this is really the goal that patients really aspire to achieve.
Thank you. So the second question, just for the whole panel here, although it really focuses on Tony and Maxine's presentation. So it seems like PBRM1 mutations were associated with benefit to immune therapy in the second line, but not in the front line. And so why is that? And in the second line studies, did you use frontline biopsies, and how representative were those biopsies of what the tumor was like when you were actually treating the patient? Could the VEGF receptor TKI have enriched for a population that had PBRM1 mutations that you missed in the frontline treatment?
Absolutely. I mean, these are archival tumor. This is, absolute. There is no explanation that come to mind except that how the tumor microenvironment or the tumor composition in general is different pre and post TKI. We are working on that. We have cohorts of patients with primary and metastases without treatment in between and with VEGF TKI, and we want to see what is the difference. But I want to re-emphasize that despite this somewhat validated, this is weak at best, modest at best, weak to modest in between. So there is an association. This will never be, if the data stands like this, used in clinical practice. And I think, Mike, the other thing is we are a bit past nivolumab second-line therapy, at least in the US post-TKI. This is really not the standard unless you can't use ipilimumab or, other TKIs.
So just, thanks, Tony. So I think the message is, I think if you're going to do biomarker studies, you have to pick the right endpoint, and you have to have the right samples to start out with. And then we can debate what are those biomarkers. But Brian, just one quick question. How are we going to be able to choose who should get Axi-Pembro versus Ipi-Nivo based on the data?
Thank you for that quick and easy. I mean, I guess, so I'm sort of, here's my opinion. I mean, they're both active regimens. I'm coming to the opinion that VEGF inhibition, PD-L1 inhibition, PD-1, PD-L1 inhibition, and CTLA-4 inhibition are probably all important for kidney cancer patients. And as I sort of alluded to, I mean, how do we intensify or de-intensify those three axes, so to speak, across patients? That's what we don't know. So how's it going to be decided? It's going to be more practical stuff, right? I mean, they're both active, they both have survival benefits, et cetera. I think it's going to be a more practical toxicity. Do you want an all-IV regimen, et cetera? I think right now that's how it's going to be decided. If you're asking biologically, the answer is I don't know. I mean, there are studies going on, from BMS and Merck to get at, the biology of responders, however we want to define that, but we're a ways from that being clinically useful.
Hey, does this work? Okay, great. So I would just like to point out, Brian, that was a great talk, and I think that you did a nice job talking about those signatures and how they might correlate with response, but those were not biomarkers. That was a signature, right? And it's like a long way from a signature to a biomarker. You got to choose the right number of genes, you got to generate a CLIA-certified assay, and then you theoretically have to prospectively validate it, right? So like designing a trial based on entry, based on these signatures for which we don't really have a validated assay yet is kind of a little bit, and I thought your idea was good, right? But I just couldn't vote for it because, again, a signature is not a biomarker, right?
It's conceptual, not practical. But I don't disagree with you.
We do want something simpler. When we took the RNA-seq from 150 and applied it, there was 75% only concordance in a larger study. What does it mean? Do you need to go back and get our signature? Then someone will come up with a 1,500 patient and they will bring 3-4 gene more, 3-4 gene less.
But that's okay. I mean, so in breast cancer, right, there's two validated biomarkers, mammoprint and basically Oncotype DX. They have different signatures, right? People use them in different settings. So that's okay. It's just that it has to be a CLIA validated assay that's actually prospectively validated to be a biomarker, right?
Yeah.
So I'll just say that, I mean, those gene signatures were validated in 151. So they were developed in 150 and validated in 151. Whether it ever gets a CLIA certification in clinical use is a more complex question based on the regimen and regulatory filing, et cetera.
Yes, 100% agree with Chuck's comments. And I look at these, I listened, watched Tony present the Javelin biomarker at ASCO, and then Paul presenting it again. I think what I get is you have 700,000 patients, 1,500 patients, you do all this expedition, and you come up with a signature that doesn't really, at the end, help us select which patient will get avelumab axi, or axi alone, or sunitinib alone, and then you'll do the same analysis for pembro-axi, Keynote 426. You'll get another set of genes that is, maybe there is some overlap, maybe there is totally different. And just this, I can't escape the notion that this is, if you have a lot of patients and you have a statistician who works for your company, you torture or massage the data, the statistics, and you get the results you want. At the end, it doesn't help patients. How can I tell a patient in the clinic, face-to-face, here is a signature that's going to help me give you this or the other? I don't think it does. It makes good presentation at a meeting on the podium. It makes a nice paper, but does not really advance the field and does not help patients.
I just switched to prostate cancer research.
So, Nizar, I don't disagree with you about clinical utility, right? That's hard to argue, right? But I think what these signatures have done have taught us about the biology of the disease, right? So I think the advances in understanding angiogenesis and inflammatory and metabolic, I mean, those are fundamental that will help patients as we develop new drugs and approaches. But of course, you're right. I mean, that's hard to argue. It doesn't mean it's not possible.
But I also think there's work that still needs to be done there because we haven't actually run a prospective trial on the basis of any single marker or signature yet. And there may still be potential benefit or utility there that hasn't been proven yet. But retrospective analysis can be risky for all of the reasons that you highlight.
Yeah, we certainly have to keep trying. The question that I would have is actually for the pathologists on the stage and in the room actually is there are all these technologies that are coming up where obviously your scope of work and the way you approach tissues is now getting far away from the H&E staining and just looking under the microscope. And we have talked about multiparametric staining, by the way, congratulations, beautiful work done. But also looking at technologies with, let's say, mass spectroscopy, localized on slides, looking at hundreds of protein markers on these things, being at institutions thinking about how do we invest in these technologies in the future. Is there some kind of task force that kind of brings pathologists together and talk about this and decide, okay, these are the platforms we want to invest in, these are the platforms that we want to deploy across clinical trials or some more comparison. What's going on in that respect?
Well, I think there is a lot of interest in multi-parametric analysis and spatial analysis, of course. I mean, this is going to be, and the technologies are getting there. The mass spec-based, tissue analysis are available, and we are starting to work with this, at least at our institution, with this technology. However, as I mentioned yesterday, the throughput is extremely low. I mean, it's something that it's not yet prime time for analyzing hundreds of samples in the context of clinical trials. So I think that can be utilized, in parallel with the single-cell sequencing. It's more of a discovery at this point effort that you can do some, pilot study or an analysis of responder versus not responder to try to discover something that you can bring to the more clinical, CLIA certified setting in trying to develop a biomarker that you can use in the clinic. With respect to CLIA certified applications, like at the Brigham, at least we have started to do immune profiling. So just do a multiplex IF CLIA certified assay to start, interrogating tumors, not only in kidney cancer, but obviously in all different tumor types. That's a more, mundane exercise, but I think it can, certainly provide very many information. So I think, from a perspective of a pathologist, I'm more focused on, hypothesis-driven work. We have few markers that we can assessing, the target of the drug and the ligand, things like this in tissue. It's possible to enrich. I understand that there is a lot, a long way to go, but if we can start just enriching for the population that can respond, just doing something that can be easily implemented in the clinic. So that's the hope. I'm not sure that, I think both discovery and much more clinical assay need to be continued to develop in parallel. Bill.
Jim, you have a question?
I think Bill was first.
Go ahead, Bill. Go ahead, Bill.
So a couple of
points. So first is Arlene Sharp has just published, but I know of unpublished data from at least one other group, that when you inhibit the VEGF receptor on the T cell, it becomes a much more effective T cell when it comes to tumor cell eradication. So this dichotomy between immunotherapy and quote-unquote angiogenesis inhibitor may get blurred. Mike even suggesting that for many years, but I think now there's more and more data to support that. So that's number one. Number two, in my mind, at least with respect to patient selection, the most useful biomarkers are digital in two senses of the word. They're digital in their measurement, like you do have this mutation or you don't. And then depending on that biomarker, you are or are not going to respond to this agent. Everything I've seen so far here is very analog-y, both in the measurement and the interpretation. So I think for biological discovery, this is fine. I'm very skeptical whether this is going to get us to a point where we can really make meaningful clinical patient selection, as was mentioned earlier. And then I would add on top of that two things. First of all, biomarker discovery, it's always a moving target because it's a biomarker for the therapy today. By the time you validate the biomarker, hopefully we're into some new therapies, and so you're sort of always playing catch up. That's number one. Number two, I'm really fearful that these biomarkers are going to be used as an excuse to get the very last mile of benefit from mono therapy and some of these early two-drug combinations, when in fact the end game is clearly to get to 3 or 4 drugs that have complementary or alternative mechanisms of action. And so, we can maybe eke a little bit more benefit out of sunitinib, et cetera, but let's just get to the future. And so I think some of our resources should really be going there as opposed to biomarkers. So that's I'm the contrarian, though. I'm the designated contrarian. So I know there's a cottage industry that does it, but
That's good.
So, Paul, a couple of questions for you. One is, did I hear you say that you tested the in-motion signatures in the atezo-bev trial and that you found it similarly predictive? And if so, I would like to know how much value added there was from the second signature. And then the second question is, I thought the approach that you described of eliminating all of the genes where you didn't see a lot of variation and then ended up with 4,000 genes and then looking at the ones that varied the most and so on was a good approach. But wouldn't it make sense also to have a supervised approach and look for the genes that might be most predictive in one setting and another?
So we've taken both approaches, but let's start with the Emotion 150 signatures. So I should clarify, and I apologize, this slide was deleted, but the T effector signature enriched for response in the combination arm. However, the combination of the T effector signature and the myeloid inflamed high signature, which favored the atezo-bev combination, actually didn't favor the immuno combination in our trials, so there was a difference there. The angiogenesis signature did favor the sunitinib arm, as you might expect, as did ours. But to the second question about proceeding to develop signatures in a supervised way, we've also examined that and identified signatures with some overlap as well as discrete signatures, depending upon exactly which questions we're trying to address. The signature that I showed you here was probably, to respond to the previous comment, the least manipulated, it was essentially the genes which were most significantly associated with response, and that was the filter. There was no picking or choosing, and so we felt that was probably the least biased approach to start with, but we have looked at different subsets and tried to derive signatures for different patient subsets. In some cases have been successful, in others we weren't able to do it. So there's potential there. I think there's more work to be done.
Okay. There's some other questions here, but I think you can ask directly to the speakers because I think it's time for lunch, or the photo and then lunch.
Okay. Would